Welcome to the Immuno-Oncology webinar first session. Today's topic is Immuno or Bust, is Immuno-Oncology in a Bubble? Our presenters today are Robert Young, Senior Scientific Analyst from Biomed Tracker, Mandy Jackson, the West Coast Editor for Scrip Intelligence, and Dr. Sandeep Patel, an Assistant Professor at UC San Diego Moores Cancer Center. Robert Yang has been a Senior Scientific Analyst overseeing Biomed Tracker's oncology coverage for the past 5 years. Before joining Informa, Robert was a cell biologist who earned his PhD from Stanford University before working as a postdoctoral researcher at Berkeley and a staff scientist at the Sanford Burnham Institute specializing in cytoskeletal dynamics. Mandy Jackson is the U.S. West Coast Editor for Scrip Intelligence. Her focus is U.S. biotech and pharma, including financial, deal-making, and clinical trial news. Mandy has been a business news reporter for 15 years, covering biotech and pharma companies, biotech law, and commercial real estate. She's based in San Diego, California. Our guest today, Sandeep Patel, is the Assistant Professor, Cancer Immunotherapy Program, Experimental Therapeutics, Thoracic Oncology, Division of Hematology and Oncology, and Center for Personalized Cancer Therapy at UC San Diego Moores Cancer Center. Dr. Patel is an expert on cancer immunotherapy and early phase clinical trials involving immunotherapy across all types of cancer. Dr. Patel's research focuses on developing biomarkers for personalized immunotherapy to better determine which combination of therapies works best for the individual. Dr. Patel earned his medical degree at Baylor College of Medicine while performing research at MD Anderson Cancer Center. He completed a residency in internal medicine at UCLA Medical Center. He completed a fellowship in medical oncology and hematology at Duke University Medical Center. He is triple board certified in internal medicine, medical oncology, and hematology. Now, we will get started. Robert, Mandy, let's begin.
Hi, everyone. Well, immuno-oncology obviously is the hottest property right now in biopharma based on the response rates and overall survival benefits that we've seen to date. The first round of immunotherapies, Gendrion's Provenge for prostate cancer and Bristol-Myers Squibb's Yervoy or ipilimumab, have had mixed results in the commercial market, but obviously the industry has high hopes for PD-1 inhibitors, the newest class to reach the market. That's Opdivo or nivolumab from Bristol-Myers and Merck's Keytruda or pembrolizumab. PD-1 and PD-L1 inhibitors are projected to generate about $40 billion in annual sales at peak, with estimates of $7 billion in lung cancer alone. That's why Roche, AstraZeneca, Pfizer, and others are playing catch-up in development. Right now, there are more than 100 clinical trials underway testing combinations of PD-1 inhibitors with chemotherapy and other targeted cancer drugs. Immunotherapies are entering into their own, or other companies are entering into their own monotherapy and combination studies and making big investments in their own portfolios and partnerships with others. Celgene paid $1 billion up front to partner with the CAR-T developer Juno Therapeutics, and Sanofi paid $650 million up front to partner with Regeneron. And then Pfizer paid $850 million up front to work with Merck Serono. So with the rush to immuno-oncology, we've been thinking, is there a bubble that could burst at some point? But our guest, Dr. Patel, has worked with these agents in clinical trials, and based on the unprecedented efficacy he's seen, he doesn't think so. So Dr. Patel, what makes you think that this isn't hype, or the hype that we are hearing is worth it? Why are these drugs so incredible to everyone?
Yeah, so I think a couple points. I think when we talk about bubbles, that's really a valuation question financially, and so that's not my field of expertise. I'm more in the clinical science realm, and so if we're talking about scientific bubble, is there a lot of hype? I would actually say no. And so having seen the development of these agents and having seen patients respond, what's particularly impressive is the durability of response. Those patients that respond can respond for maybe 2-3 times what you expect from conventional agents, with some responses lasting years. If you look at some of the earliest trials of ipilimumab, Yervoy in melanoma, there's some survivors with metastatic melanoma who are almost a decade out on therapy. And so it's that level of response that's really changed the game compared to what we're used to seeing, even with very effective targeted therapies. And so from a scientific standpoint and a clinical standpoint, taking care of patients, I actually think we're just getting started in terms of immuno-oncology, and as we better understand the pertinent biomarkers that are required to analyze for each individual to come up with kind of a personalized immunotherapeutic combination, I think we'll be able to push the bar further and get more and more patients to respond for longer and longer. So from a scientific standpoint, I think we're just getting started.
So if I could follow up on that, because you brought up Yervoy. Let me just play devil's advocate and say, is this entire interest in immunotherapies, or is all the excitement about immunotherapies, as we talk about a bubble, really just driven by a single successful target, PD-1s? Because, as you mentioned with Yervoy, and before that we had Provenge, those were also immunotherapies. They were also generally successful. But if those were the only ones that were successful and we didn't have PD-1s, we wouldn't be talking about this excitement with immunotherapies.
I think that's a fair statement, especially in the sense that when you look at sipuleucel T, Provenge efficacy, which never really had a major benefit outside of overall survival. We weren't able to see benefits in terms of biochemical response, PSAs, and the patient population was very select, patients without painful bone metastasis, a very self-selected population. When you look at the responses with the PD-1 agents and anti-PD-L1 agents in clinical trials, you definitely see responses in terms of tumor shrinkage, and the durability of those responses is particularly impressive. I think when you talk about the combination strategies, I think that's really where the PD-1s and anti-PD-L1s have been particularly effective, because their side effect profile is amenable to combinations. In fact, the most toxic combination that I'm aware of in the immuno-oncology space is the combination of CTLA-4 with PD-1, PD-L1. So, CTLA-4 agents are centrally acting agents. They activate a lot of T cells very briskly. They're very good agents for the right patient population. I think which patients need the combination versus which patients can do well on a monotherapy, such as PD-1 or PD-L1, we're still learning about. And there's some data that if you already express PD-L1 in your tumor, your benefit from adding CTLA-4 may not be as much. And I really think so those kinds of biomarker decisions really help guide which combinations may make sense for a given patient.
But again, so as you're saying, the baseline here is the PD-1, and because their side effect profile is so amenable to combination. I guess I would argue, again, it's the PD-1s that are so exciting. But any other combination, either with a targeted therapy or another immunotherapy, if they're not as, if their safety profile is not as terrific as PD-1, they're going to bring whatever AEs to the table that they have in combination, as you were saying with Yervoy. So we're still just talking really about PD-1s being the backbone of every combination.
And I think that's marketplace. And if you look at the amount of money being spent by companies to acquire PD-1s or PD-L1s when they did not have them in their pipeline, I think you mentioned Pfizer in particular already. And if you look at just the amount of money and the number of clinical trials invested in PD-1 or PD-L1 development, as well as combinations based on that backbone, I'd be in agreement that I think in the immune checkpoint space, PD-1s and PD-L1s are the base from which we're building off combinations with all the other novel targets that are being looked at. When you look at portfolios that include, IDO and GITR and CSF1R, we actually, talked 2 weeks ago, I think, on the phone. And since we've talked, there have been, I think, 2 new approvals in melanoma by the FDA, and one company, 5 Prime, has a CSF1R compound that I think was acquired by BMS. And so even within 2 weeks since we last talked, it's a very dynamic field that's changed. And based on early efficacy data, companies are making big expenditures to bolster their combinatorial immuno-oncology portfolio based on safety data and the ability to avoid toxic adverse events, as you mentioned, but also just efficacy data and efficacy signals that they're seeing in early phase. That said, not all PD-1 combinations are necessarily non-toxic. I mentioned the CTLA-4-based combinations, which is now FDA approved in melanoma with really phenomenal response rates. I mean, the 1 year of overall survival of metastatic melanoma, really over 80%, and some of those patients having durable responses, as I mentioned, some lasting years. Other combinations that one would expect not to be as toxic have proven so. So AZD9291, which is a third-generation EGFR inhibitor, which is developed for non-small cell lung cancer, that combination with durvalumab, which is AZD's MedImmune's PD-L1 inhibitor, at least in an Asian population, had almost a quarter of the patients develop pneumonitis. And so one would expect, well, we have two seemingly non-toxic agents as monotherapy and historically amenable to combination strategies with a surprising toxicity as well. And so even though overall PD-1 and PD-L1 are more amenable to combinatorial strategies, we still need to actually do the clinical trials to figure out if that holds true.
Yeah, I think just, if I could just jump in with my own take as a former, academic researcher is, I think one of the tendencies, one has to be cautious because often a single proof of principle tends to get, is sufficient in the academic realm, but in the marketplace where we're talking, there still is a considerable risk with some of these up and coming immunotherapies, and because one immunotherapy or one checkpoint really has been so successful, it's been shown such a terrific efficacy and safety profile. I think there is a strong tendency to expand this to all immunotherapies and say, okay, we've shown now finally that immunotherapies really can work. But again, doesn't mean that other immunotherapies won't have surprising, toxicity, or even in combination, as you're saying.
Exactly.
But given that sort of caution, you just mentioned a few of the interesting targets. I mean, can you talk about what you see as the most promising targets besides PD-1s, other checkpoints, or even other unrelated checkpoints?
Yes, I think, in terms of the combination strategies, PD-1 and PD-L1, the publicly announced trials, we're waiting on results, but there's several companies with agents in various immuno-oncology spaces. IDO inhibitors, for example, multiple companies are pursuing development there. 4-1BB, GITR, you look at OX40 programs, CSF1R, as I mentioned, is another program that seems quite promising. And so, right now, the bulk of immunotherapies and immune checkpoint blockade is really related to blocking the brake on what we think is the effector T cells, the main mechanism of action. But when we think about cancer, cancer does a multitude of things to the immune microenvironment in terms of suppressing dendritic cells, M2 macrophages, regulatory T cells, and infiltrating immunosuppressive B cells. So there are a variety of immune cells and axes that are perturbed in a tumor microenvironment, and these are the ones that, if we have a better understanding of each individual's tumor immune microenvironment, can come up with a rational strategy for targeting them for each individual patient. This is kind of the personalized medicine revolution that we're talking about. And I think, secondly, really the cellular therapy space. We talk about the CD19 CARs in ALL, CLL, and diffuse large B-cell lymphoma, which have really transformed that field in particular. And it's hard to imagine patients with these diseases in a year or two won't be receiving these as a standard of care. I mean, the efficacy data really is phenomenal in the post-transplant setting. And so I think even beyond immune checkpoint blockade, the cellular therapy space, and whether as usual, the malignant hematology space is a bellwether for what's going to happen in the solid tumor space. We're hoping to replicate those successes as well.
Okay. Mandy, did you want to?
Yeah, I wondered, can you talk a little bit about how CAR T therapies fit into this case, because they are experiencing some pretty phenomenal response rates with those, and have heard some really great success stories in these early stage studies that have been done. They're primarily phase 1, phase 2, but it's a different type of therapy, the cell therapy, as opposed to a monoclonal antibody. So how do you see the CAR T fitting in and becoming successful commercially, given that it's just a different kind of therapy and it will be administered in a different way?
Yeah, I think it's a great question. I think when you look at the distribution of and the ability to administer monoclonal antibodies, we've done this for almost a decade now in the oncology space, and so the switch from giving trastuzumab or rituximab to giving nivolumab or pembrolizumab is not a major leap of faith for most. I think when you talk about the CAR space, the CD19 CAR T cell space in particular, to date and probably for the foreseeable future, these are really going to be restricted to transplant centers, places you otherwise would get a bone marrow transplant at. Why? Because some of the toxicities related to that really require intense inpatient monitoring. There is this cytokine release syndrome that can develop when they're getting infusions. And so really, I think the places that currently do bone marrow transplants will be the places that patients will be referred to for CAR T cells. That said, I think when you look at the in light of that toxicity, we look at the efficacy of these agents in those malignancies, ALL, CLL, and some of the lymphomas, some of the preliminary data that's been presented, really just phenomenal data where patients can have already had a transplant, either autologous or even allogeneic, and can receive CD19-directed CAR therapy and have response rates of 70, 80% in a post-transplant setting. Just really phenomenal data. And these responses can be durable, and you can detect persistence of these T cells in the blood years out even. And so we're definitely, it's a whole new way of thinking. Of course, the process of generating a CAR T cell is quite burdensome. In fact, we're talking about Provenge and Stipulosaltine. In fact, a lot of the pheresis procedures and lentiviral transfection steps are somewhat reminiscent of that product as well. And in fact, I think a lot of that infrastructure has been repurposed for CAR T cell development. And so there are a lot of players in that space. But I think the CD19 CAR T cell is truly a success story, and it's probably the first of others to come as these therapies are eventually studied in the solid tumor space. There is the major caveat that we know we can eradicate a B-cell population for long periods of time. That's why rituximab maintenance is so effective. Whether we're able to eradicate a solid tumor antigen at that level without causing toxicity, in some of the early phase trials, there have been some severe toxic events, and so there are going to be some cellular engineering design challenges that will have to be overcome, and whether these are scalable ultimately is going to be a major question, because at the current time, these therapies are not scalable, and it's very intensive to make a therapy for a given patient, even though we're all reprogramming their T cells to fixate on a given target.
So I think that's interesting because, so just to sort of summarize what you were talking about, you think the efficacy, because there is that significant toxicity, cytokine release syndrome associated with CAR T therapies, but you think the efficacy, it sounds like, is so tremendous that that those AEs are manageable or would be willing to be tolerated.
Yeah, absolutely. I think, and definitely when you compare it to transplant, which we've, these agents are going to move more into the frontline setting, and so you can very much imagine these agents with higher efficacy and better toxicity than the actual transplant process itself being favored over that. And then in terms of frontline versus chemotherapy and things like that, that's probably the next step of figuring out how these work and whether the durability in certain diseases is worth the toxicity.
So you think that, right, so how do you feel that would compare with chemotherapy again, because they have to go to large academic or transplant centers for this therapy? Is the efficacy, at least currently, suggesting that it would be strong enough to become the first frontline therapy, given its difficulty in both manufacturing and delivering these therapies?
Yeah, I think there's equipoise in terms of the chemotherapy regimens for ALL and CLL are very toxic, and so I think and depending on the disease, the durability of those responses is variable. But I do think in the cellular therapy space, I can easily imagine clinical trials looking at first-line therapy for CLL, for example, eventually, though CLL has had a variety of other agents, such as, ibrutinib, the PI3 kinase inhibitors, that really have changed the game. So it's one of those things where in these spaces, in the first-line setting, there's so many good options, how you sequence them for a given patient, and frankly, how long those responses are. it's going to be an individual decision that the physician has with the patient, but I can easily see these therapies moving more into a frontline space eventually, especially as we get better at managing a toxicity profile. I think going back to ipilimumab Yervoy, that's the classic example. When the early phase trials were done, people really didn't appreciate the colitis and the diarrhea and the risk that presents to patients in terms of bowel perforation. But I think now, in 2015, the physicians who've been giving these agents for so long are much more adept at managing these side effects. And if you look at the adverse event rate, it's much lower. And it's because people have understood the side effects. So I think CAR T-cell therapies are the same way. I think they have a very intense side effect profile. Institutions are getting better at managing them. And because of this, I think the ability to move these into the earlier line setting is feasible, though we're probably a couple years away from that. But really, if the responses are as durable in the refractory setting, in the first line setting as they are in the refractory setting, then one can imagine scenarios in which younger patients, for example, may preferentially pick a cellular therapy approach as opposed to taking a pill for longer periods of time, and that may actually work out to be cost effective for those patients as well, in addition to being medically very effective.
Well, you mentioned the potential push of CAR T therapies into solid tumors. Can you envision a scenario where they become a preferred therapy, or the efficacy is high enough to become preferred therapy for solid tumors, given the difficulties in administering CAR T?
Yeah, so I'm making a little bit of a leap of faith by talking about early line CAR T cell therapy, even in the lymphoma space. So I think 2 years from now, it may not be so hard to say. I think in the solid tumor space, the real issue has been what's called off-tumor on-target toxicity, meaning a lot of the antigens that we try to target with CAR T cells in the solid tumor space, HER2, for example, mesothelin for active trials, these are antigens that are ubiquitously expressed on normal tissues as well. And so on the one hand, we can essentially totally ablate CD19, and there are some rare infections you can get like PML and things like that from doing that. When you try to take a solid tumor antigen that's either cross-reactive or expressed on other normal tissues, that toxicity can be very problematic given the number of T cells that are being infused. And so there are design challenges to that once solved will probably make the ability to do solid tumor CAR T cell therapy more amenable. But at the current time, it is very difficult in the solid tumor space. There are a handful of active trials looking at this, for which we'll probably see some data at ASCO, I imagine, this summer. But I think in terms of solid tumor space, there are some design challenges that need to be solved before widespread use can be more likely used in a community setting or a widespread setting than kind of in an intense academic environment that really, in terms of solid tumor space, only a couple institutions are currently doing clinical trials on.
Okay.
Well, how do you see the agents that are in trials now and that are in the market? You know, we have a couple PD-1 inhibitors in the market. How do you see the impact on other therapies that are either in development or in the market? I mean, Yervoy seems like a prime example. Bristol Myers has been saying, or has said on their last earnings conference call, that Yervoy sales are down primarily because Opdivo's in the market. Although that may change once people start using those two in combination. And then you've had Celgene saying that their Abraxane sales took a hit in lung cancer because of the PD-1 inhibitors. So it seems like it's been a rather fast shift to have that kind of impact. I mean, do you see that continuing and having an impact on some of these therapies that were great, and now we've got these other agents that seem so much better? I mean, it's kind of an amazing uptake in the market already.
It really is, and even kind of anecdotally, you know, at our institution and you know, my colleagues, the uptake of these agents has been really extremely rapid. There's a high comfort level with these agents now because so many institutions did the clinical trials in development. So you know, when the drugs you have a better understanding of their side effect profile, and you see the efficacy in front of your eyes, and you see people respond, you know, for months, years even on some of these therapies. It really is telling, and so I do agree that I think the marketplace is going to be altered by these agents, and especially as we look forward, kind of combinations of these agents. I do think the Ipi-Nivo combination is highly efficacious in metastatic melanoma when you look at the data, especially if the patient does not express PD-L1 IHC as a biomarker. Those patients definitely benefit from combinatorial strategies. And so I don't know how Ipi will go forward, but I can say in the current scheme, there are definitely melanoma patients that benefit from that combination. And there has been some lung data with alternative dosing that was presented at World Lung of the Ipi-Nivo combination that look promising as well. And so these are definitely things we'll be staying tuned for in terms of the combination versus monotherapy. I think in terms of kind of your traditional cytotoxic chemotherapy agents, some of these trials are combinations, meaning they'll combine chemotherapy with PD-1 or PD-L1 inhibitors, and some are versus, meaning in the same way patients with EGFR mutations or ALK or ROS1 in the lung cancer space, or BRAF mutations, for example, in the melanoma space, whether these patients actually can avoid chemotherapy if they express the biomarker at a high level, for example, patients who have high PD-L1 expression. And so I think these are all active trials that are ongoing, and we don't know the right answers yet, but based on the efficacy data in the refractory setting and really the amazing tolerability of these agents compared to conventional cytotoxic chemotherapy, one could imagine that if any of these trials are positive, it would really be a dynamic, it'd be even more of a sea change moving to the kind of earlier line setting for these immunotherapeutics.
So just to follow up on that, I mean, you're talking about trials looking at chemotherapy versus the PD-1, for, especially for ones who are biomarker positive. But because of the amazing tolerability, could you see a scenario where every patient, regardless of PD-L1 expression status, just basically receives PD-1 therapy as a standard, especially with the delay to onset of efficacy, right? So some of these have no effect on progression, but they have an overall survival benefit. I mean, could you see a scenario where everybody just receives these as standard therapy up front?
Well, I think right now, in terms of standard therapy up front, so the melanoma space, especially if you're BRAF negative and even for your BRAF mutation positive patients, the vast majority of the patients in the country are receiving PD-1 or PD-1 nivolumab plus ipilimumab in the frontline setting. So melanoma, the use of chemotherapy, I'm not sure the last time someone's written a prescription for dacarbazine or written chemo orders for dacarbazine in melanoma, because the number of agents, especially immuno-oncology agents, has really blossomed in that space. In the lung cancer space, in the second-line setting, currently the approvals for nivolumab are without biomarker, meaning you do not have to test necessarily for PD-L1 in either the squamous or the non-squamous space. For pembrolizumab, there is a biomarker, integral biomarker test. And so there are approvals in the refractory setting. I think in the first-line setting, if you really parse through the data, the patients who are PD-L1 IHC 0, stone cold, no expression of PD-L1, those patients actually do better on chemotherapy. And so I think the biomarker is going to help us decide what therapy makes the most sense for each individual patient. I think when you look at patients with kind of low expression of PD-L1, at least when you really parse the data out, particularly from the BMS and Merck trials, those patients with lower expression, they may benefit as much from chemo, maybe a little more, maybe a little less, but the side effect profile is substantially better. And in those patients that are biomarker positive, strong PD-L1 IHC high, benefit preferentially with PD-1 blockade. And so I think really the goal is with the PD-1 inhibitors and PD-L1 blockade is to avoid giving it to patients who actually would do better on chemotherapy. And that tends to be the patients who are truly PD-L1 0, stone cold, no expression of the biomarker. And that kind of makes sense. We've learned that in other disease types. If you give BRAF inhibitors to patients without BRAF mutations and things like that, they're not going to respond, and it makes sense because they don't have that biomarker.
But at least as much as I've understood about the biomarkers, the PD-L1 biomarkers, is there is a lot of struggle to make them more reliable. And every company has its own biomarker, and some are very prognostic, and some are not. And what I've heard other people at ASCO saying is that even though those who are PD-L1 0 expressors, there still is a fraction who do respond. And so what I'm wondering is, if because the toxicity profile is so good, even if they benefit from chemotherapy, would early treatment with PD-L1 for everybody, regardless of biomarker status, ever be warranted, and then they could receive chemotherapy on top of that if there's either early signals of no response or even just in combination with chemotherapy?
Yeah, I think it's a great question, and I could definitely imagine that scenario. The one caveat to that is the time to response with PD-1s is much slower than any of us would like in terms of monotherapy.
Which is right. Which is why I was wondering if really early treatment would be warranted for everybody, because you don't know for a long time.
And then the question is, so standardly in oncology practice in clinical trials, the CAT scans every 6 to 8 weeks. And so with the PD-L1s, oftentimes, for example, in non-squamous lung cancer, you may not know if they're working or not until you get to month 4. And that's an awful lot of time for patients with metastatic lung cancer to make a decision on whether it's working or not. Are you having what's called pseudoprogression, where the cancer is growing, or you think it's growing because the immune cells are getting into the tumor, making it look bigger, and are they going to respond later or not? So this has actually been a big problem in the field of trying to figure out which patients are going to benefit later on after having an initial pseudoprogression event, which is much more common in melanoma than in lung cancer, versus those that actually are progressing and you need to switch to something else. And so I definitely think what you mentioned was kind of an adaptive design, meaning a patient that doesn't respond early, they get something different in combination, whether that's a PD-1 plus another immuno-oncology compound, a targeted therapeutic agent, a chemotherapy agent. I think that design is very attractive, and I think it makes a lot of sense, and I could definitely envision what you're saying. But I think the one caveat is the amount of time we have to wait right now to figure out if a patient's going to respond to PD-1 blockade in kind of non-squamous lung cancers. That amount of time can make it quite difficult if you picked incorrectly to start with. And I think you bring up a good point that patients are PD-L1 IHC 0. Depending on the tumor type, anywhere from 8 to 10% can have a response that can be as deep or as durable. And I think that issue is a very complex one related to issues related to antibody, the staining, the biopsy, or did you get biopsy, do you have enough tissue? Kind of in the same way that you can do a biopsy for next-gen sequencing, and you can miss a mutation because you just don't have enough cellularity of the tumor. Kind of the parallel to that is if you don't get enough of the tumor microenvironment, the immune microenvironment on your biopsy big enough, you get a false negative PD-L1. Now, I'd actually make the argument that a false negative PD-L1 IHC is the worst possible outcome in terms of biomarkers. You otherwise have therapy that's very efficacious that a patient's not going to get. So even in our use of these agents in the CLIA space, in terms of trying to better predict which patients are going to respond or not, in my own head, when a patient's PD-L1 positive, I feel very comfortable. When the PD-L1 negative, exactly as you said, I always wonder, how good was the biopsy? Did it miss? Are there other tumors that we didn't biopsy PD-L1 positive? And so there's always that in the back of your mind.
Right. Mandy?
Well, what other biomarkers are you seeing that may be better predictors of response for some of these agents?
Yeah, I think, so right now when we talk about immunohistochemistry and PD-L1 protein expression, I think that's the most advanced one. But I think, as we've kind of alluded here, that's definitely not the only one, or arguably even the best one. Another key characteristic of all these therapies is you're basically unleashing T cells that are antigen specific. And so if a tumor has a high neoantigen burden, you're statistically much more likely to respond to immune checkpoint blockade. And so that manifests in basically two ways. One is essentially the denominator, just the sheer number of mutations. So patients, for example, with GI cancers or gynecologic malignancies with microsatellite instability, for example, those patients have preferential response to PD-1 blockade because of their high mutational burden. You can look also at specific antigens that are immunogenic, which is really where the field's gone, because that's actually how you come up with personalized vaccine strategies. Historically, cancer vaccines have not done very well, A, because of an immunosuppressive microenvironment, but B, whether we're vaccinating against something that's immunogenic for that particular person and that will lead to anti-cancer immunity, the ability to design personalized vaccines really hasn't been available until, frankly, the past couple years. And so those strategies are being developed as well. But that really goes back to the question of, besides protein level, there are DNA changes that may portend response, and one is just having, an immunogenic neoantigen, for which there's several publications in the ipilimumab space and tremelimumab space that describe that. And then the other thing in particular that there is some data for is transcription at the RNA level. So in particular, looking for type 1 interferon gamma signatures or MHC1 presentation signatures in the tumor microenvironment that indicate there's kind of an inflamed transcriptome that can be unleashed if you gave the right combination of immunotherapeutics, such as PD-1, PD-L1, or even some of these novel agents that I'm mentioning. And so I think really PD-L1 IHC is the base, and it's what's clinically and CLIA approved. But it's hard to imagine a few years from now that it won't be a constellation of biomarkers that predicts which patient will require which particular immunotherapeutic combination and maximize their chance of response.
When you're talking about with the biomarkers, I know your sort of specialty is looking across tumor types for more personalized medicine, but it does seem like there are some tumor types that are responding better, especially to PD-1 than others. And so are there other tumor types besides lung, melanoma, maybe renal cell, that meet some of the criteria you're talking about more generally? Because I know you're looking at specific patients and anyone who might need a constellation of biomarkers, but are there tumor types that already sort of generally meet these criteria more than others?
Yeah, it's a great question. I think so. Hodgkin's lymphoma is probably the classic example. So I mentioned kind of DNA, RNA, and protein level changes that portend response to PD-L1. And so Hodgkin's lymphoma, in particular, has an amplification of the 9p chromosome. The 9p chromosome contains PD-L1, PD-L2, and actually JAK2 in particular. And so these three are amplified in patients with nodular sclerosing Hodgkin's lymphoma. And so there was a trial in which patients who had already had a transplant or had received brentuximab, which is a CD30 antibody drug conjugate, who were refractory to those very effective therapies, were treated, and the response rates were upwards of 85–90%, I think, in that setting. And so this goes back to the point there was a genomic event that leads to enhanced transcription of PD-L1 and enhanced expression of PD-L1 on the tumor surface. So you can check at any one of those levels PD-L1, but that's kind of a unique mechanism. Other cancers, and there's a famous slide they showed at ASCO probably on every single immunotherapy presentation back in 2014, was kind of the mutational burden by histology. And so when you look in terms of the fewest mutations per megabase, those were kind of pediatric diseases, ALL, neuroblastoma. Makes sense from that standpoint. On the far right side, there are tumors with high mutational burden. These are melanoma, squamous cell lung cancer, non-squamous non-small cell lung cancer, bladder cancer. And so these should all sound familiar because these are actually all the histologies for which immune checkpoint blockade is already FDA approved, or bladder cancer looks very promising and it's one of the other histologies on deck. I think head and neck was very high up on that list, head and neck cancers in particular. And I think, one is head and neck cancer. There's an epidemic of HPV positive head and neck cancer, which is present in never smokers, or often non-smokers, non-tobacco users. And so whether these strategies are particularly amenable for these virally mediated cancers as well, I think is another potential histology. But I think you've named some of the ones that have already been approved, melanoma, non-small cell lung cancer, renal cell is probably on deck. But I think when you look at some of these others, bladder, head and neck data, and you look at some of the efficacy there, Hodgkin's lymphoma, I think there's likely some new approvals there as well in those histologies. And it's very likely that these are monotherapy treatments. And so if you pick the right backbone of either targeted immuno-oncologic or a combination of immuno-oncologics, you may kind of increase the number of histologies that are amenable. But really, it does seem, when you look at the data and the response rates and the biomarkers, that the histology necessarily doesn't matter, meaning what organ the cancer starts in. It's really kind of the associated either genomic or proteomic characteristics that determine whether patients respond to immunotherapy or not. And it kind of makes sense from how the immune system works.
Right. So do you think there's going to be, I mean, you're talking about the mutational burden being related to the efficacy of PD-1s. So what you're saying is it probably will carry true for other checkpoints in development, the OX40s and everything. So it would probably be a similar pattern. They might target the same or have the same efficacy pattern across solid tumors or across.
It would make sense with the T-cell directed therapies, especially immune checkpoint blockade or what you mentioned, kind of these TNF super receptor family agonists, kind of the accelerators of the T-cell, that the T-cells in the microenvironment, if there are a lot of mutations and you have a T-cell that is programmed to hit those mutations kind of at birth, that the more mutations you have, the more likely you are to generate that immune response against the tumor. I think that it's likely to hold true for all kind of the effector T-cell directed therapies. It would be my guess.
Okay.
Well, how do you see combination therapies coming into play? I mean, I know there are a lot of clinical trials underway. Are we concerned, and I think we alluded to this earlier, but are we concerned about the toxicities that we see when we combine agents? I mean, the PD-1 looks good in terms of toxicity compared to chemotherapy, but when it's combined with other agents, is there a concern about getting the toxicities that just aren't worth combining two agents that are costly and make the patient feel terrible?
Yeah, I think so. There are a couple ways to think about the combinations. One is kind of IO combinations, of which the FDA-approved combination of metastatic melanoma, ipilimumab and nivolumab, is one in which the grade 3-4 toxicity rate is pretty high. It's about 10-15%. Often they get diarrhea, rash, endocrinopathies, liver dysfunction. So it's a very toxic therapy overall. That said, the efficacy of the combination, especially in the PD-L1 negative IHC patients, is really phenomenal. And so it's one of the cases where even though the toxicity bar is a little higher, the efficacy bar is so high that we feel very comfortable giving that combination to the appropriate patient. I think sometimes you have unexpected toxicities from combinations, and I mentioned earlier AZD9291 plus durvalumab. These drugs are both very clean individually, and are very amenable to combinatorial strategies. And so I think a lot of us were surprised when that combination turned out to be toxic, especially because there have been data already presented about combining erlotinib with nivolumab, for example, at ASCO. So I think that's why we have to do clinical trials, because even if in monotherapy or even in clinical experience, and even if in the models things look good, ultimately, until you do the trial, you don't know. And I think I would say most of the trials in immuno-oncology that aren't kind of phase 1 testing for the dose of a new agent really are evaluating combinatorial strategies. And I think when you look at those combinations, the published data seem to suggest that they're well tolerated with a potential signal for enhanced efficacy. And so I think that's really driven kind of all the major players in oncology to have a PD-1 or PD-L1 from which they can test their internal pipeline in terms of combinatorial strategies. And B, it's really driven kind of the science forward and trying to figure out, well, which combination makes the most sense for which patient. Because not everyone probably needs, 2 or 3 drugs as part of their therapy. Depending on what the patient's tumor looks like, you may be able to finesse, a doublet or even monotherapy that works very well for that patient.
Are there particular combinations that you see coming up that you could just look forward or are particularly promising?
Yeah, I mean, I think even since we last talked, BMS has acquired CSF1R compound from 5'. I think it's a very interesting mechanism, and we'll see what the published data shows. But the fact that that acquisition was made kind of at an early point, at least to me, and as a lay observer, signal that the data must look good. I was not involved with that study. I think when you look at some of the other studies that at least have had posters or some preliminary presentation at a variety of conferences, some of the combinations looking at IDO, OX40, GITR, 4-1BB, LAG3, TIM3, these combinations with PD-1 have looked very promising from the limited data that have been shown and well tolerated as well. And so I think this is one of the spaces that's moving so rapidly that I think as we look at kind of the conference schedule, every conference is having more and more of the data presented. And I think really over the next 12 12 to 18 months, we're going to see a lot of the data formally published and orally presented, and we'll be able to better vet kind of the clinical trial data.
This might be stepping a little bit out of your comfort zone, but as we're talking about combinations, how do you feel prescribers will feel about prescribing potentially two very expensive therapies? You know, right now, if it's in combination with chemotherapy or something, that might not be a big deal. But you know, you're talking about the Yervoy Opdivo combination. You know, these are two very expensive therapies. Is there any consideration from prescribers on that?
I definitely think there is. So, you know, practically there's two levels of finances, right? One level is kind of the patient-specific finance, right? What's the copay for the patient? Is this financially burdensome for the patient? And I guess the other level is kind of the societal level. How much can society afford these medications? And I think the way, at least I personally reconcile it, is, you know, right now there are a lot of expensive medications that are given that don't work so well, that don't provide what I think in other societies they describe as value or quality-adjusted life years for that patient. But I think when you look at therapies that really move the bar, and I think when you look at these immune checkpoint blockades, these are therapies that have moved the bar. I think most of us are very comfortable giving those agents. And so when you look, there are a lot of drugs out there that we currently give that are standard cytotoxics that are quite expensive, and the responses are fleeting and the toxicity are high. And so most of us feel pretty good if we help someone, you know, even with an Ipi-Nivo combination, which is quite expensive. But if they have a dramatic response, they have their life back, and you know, they have a 2-year 80% survival from when, you know, a couple years ago that survival rate is probably what, 20 or 30%. I think those really are eye-opening, and for those we would never, you know, try to internally ration, you know, a therapy, you know, for our patients. But I think when you look at drug costs and you look at everything that's been said in the news recently and things like that, I think it is a societal level problem. But you know, kind of when I'm with my patient, my biggest concern is what are their out-of-pocket costs, and then what can I do to maximize, you know, the quality of life and quantity of life that they have. And so these agents have moved the bar in that standpoint. So I feel very comfortable using them. Most of my colleagues, most people I talk to, seem very comfortable from that standpoint as well.
Okay. I mean, one of the things I thought was really interesting at the last ASCO was the emphasis on cost.
Yeah.
And so many of the sessions, including the plenary session, had a major presentation just on getting prescribers to think about the cost, whether or not from a societal standpoint or even from an individual patient standpoint, you have to have prescribers think about how much are you moving the bar, you know, with this patient, and relative to the cost of these therapies. So I think that's something that's really becoming a, you know, really strong topic even among prescribers to think about more.
Absolutely, and I think you know, bring up a second point. There's not a lot of cost transparency out there, right? And so even at a provider level, to be able to say, I know this is exactly how much this patient is paying for this combination, that kind of data, it's not actually that, not the way our health system is set up. And so there's ambiguity to begin with, A, and B, you know, the agents that we use, you know, there's strong data for in terms of survival, quality of life, and so with those, I think most of us feel very comfortable with the IO space. But I think the other point I'd make is when we talk about these expensive combinations, not necessarily everyone needs a particular combination. Some patients may get with monotherapy. Some patients will need combination A versus combination B. And so this is really one of the areas in which the biomarker space will not only better define which patients will respond, meaning you get an efficacy benefit and also a toxicity benefit because you're not giving them drugs that don't work, but it's also a cost benefit to society because you're better selecting for the patients and picking the right therapy for them. And so I think it's one of those win-win-wins. And I would agree that where we are with the biomarker space now in 2015 with just a PD-L1 IHC, while we're in a good spot, it's not a perfect biomarker. We mentioned the PD-L1 negatives that respond, but I think as we get better at profiling each patient's tumor, and this is really kind of the personalized medicine, precision medicine revolution, I think we'll be better able to treat patients at an individual level, and I think that'll actually, in aggregate, not only help patients live longer and better, but I think it'll also be better for society in terms of cost because you're not giving them ineffective, expensive therapy.
Mandy?
Yeah, and I was just going to add to that. I suppose also maybe patients won't be going through 5 or 6 lines of therapy. They'll just need that first line.
Exactly. And so, these therapies not only move the bar in the short term, meaning they shrink tumors, but responses in many are durable. And so it's that durability of response, whether it's with cellular immunotherapy or immune checkpoint blockade or more combinations thereof, that really are exciting to all of us in the field, because kind of with the interleukin-2, which was one of the first immunotherapies developed, given in ICU, a patient would be admitted to an intensive care unit. Why would we do this? Because about 10% of patients would have a durable response lasting years, some patients up to even a decade with metastatic melanoma and renal cell carcinoma. And so the promise of some patients having metastatic disease and going for years of immunotherapy with these new agents that are not administered in an ICU setting, which are better tolerated overall, that's really the bar we're trying to move. Is we're really trying to help not only have patients respond now, but respond years in the future, even with metastatic disease. And it's a high bar, but I think with these agents and in combinations, and really rationally designed combinations based on better understanding of their tumor biology, I think we'll be able to do better and better for our patients for longer and longer.
If we're thinking about the value of these drugs, and Mandy was sort of alluding to, maybe we'll have fewer therapies, patients will have to go through fewer therapies. I think one of the characteristics of these, especially PD-1s, is the durable response. But even for patients who stop therapy, so right now, as I understand it, they're treated through progression. But even those patients who stop often have a very durable response. Do you think could you see a scenario where patients, as a more of a standard therapy with PD-1s, get a shorter duration of treatment, and basically they stop treatment much faster?
Definitely, I think that's kind of another level. So part A is selecting the right therapy for the right patient, kind of based on their tumor biology. But part B is really how much therapy do they need, and I think you've nailed it in the sense also that when you hold, so there are patients who have toxicity, you hold the therapy, they stay in remission, and you can wait till restart. And when you restart, even if they've had some growth, they'll actually retain their response. They'll regain their response. And so kind of with that understanding, the ability to do clinical trials for how many cycles of therapy does a patient need, I think that's something we have to figure out. Even with the ipi-nivo combination, remember, the majority of these patients, they're going to get 3 or 4 doses of ipilimumab based on their toxicity. And so they're not getting ipilimumab every time. They're getting their nivolumab. They're getting 4 doses, and they're getting nivolumab from there on after. And so the question is, can you do an induction course of combination and then a maintenance course for a finite amount of time, hold maintenance, watch patients, and then retreat if needed? I think these strategies are going to become more in vogue as we move forward. I totally agree.
Okay. I think we have time for maybe 1 or 2 more questions. Mandy, do you have anything?
Well, we've been talking a lot about PD-1s, but what do you think will be the next group of therapies that will either be in the market or show some of these strong or better responses?
Yeah, I think, so, as a total guess, there's a lot that could be out there, and the answer is probably going to be multiple agents in combination. I think if you look at what's on deck for the FDA, I wouldn't be surprised if in the CD19 CAR T cells, if they're the next major approval that were to happen, just given based on their efficacy and how far they along in their development cycle, pardon me. I think when you look at PD-1 and PD-L1 based combinations, the IDO inhibitors are very advanced in development. You have some LAG-3 and TIM-3 programs that are very advanced in development, and there could be some efficacy signal there that leads to approval. And then I think you're looking at kind of the next generation of T-cell directed therapy, drugs that target the OX40 pathway, GITR, 4-1BB, TGF-beta R2, CSF1R, and I'm sure I'm missing several. And so of those, what sequence those will get approved in, it's really hard to tell, but I think at least when you look at kind of the constellation of all these targets and different types of therapy, that there is an efficacy signal, and so how these play out in larger clinical trials is something we're going to spend the next 1 or 2 years really looking at, and which therapies make the most sense for which patients. But I think, in terms of the scientific aspect, and the clinical aspect of immunotherapy, I think we're not kind of at quote-unquote peak immunotherapy. I think we're really just at the base, and we're really just kind of getting started from that standpoint.
Well, maybe I can get in the last question and put you on the spot. Following up on that, is there anything that you've seen in development in the immunotherapy space that you would categorize as not promising or you think is really going to fare poorly?
Yeah, I think, so a tough question to end with.
And on the negative.
Yeah, no, but, I think some of these strategies that take advantage of kind of multiple overlapping toxicities is the biggest fear. And so when you look at some of these CTLA-4 combinations, in particular, with cytotoxic chemotherapy and things of that nature, I do have concerns about the toxicity profile. That said, even that field has evolved. And so, it used to be, well, we need to give 4 doses of CTLA-4 blockade, or we need to give a dose every 3 weeks. We figured out that we can space these drugs out further, that they may only need 1 or 2 doses, that only patients that lack an inflamed tumor microenvironment need these. And so I think giving CTLA-4-based combinations to everyone and trying to give everyone as many doses as possible, I think that era is pretty much over. But I think kind of rational use of CTLA-4, which is a very good drug, ipilimumab or tremelimumab, for patients who don't have an inflamed tumor microenvironment, or PD-L1 negative, who don't have T-cell infiltrates, I think we're going to have to be a little more judicious in our use, especially in the combinatorial space from that standpoint. That said, they're great drugs for the right patient. We just got to define who those patients are. And I think we have a better idea now than we did 1 or 2 years ago on which patients need the combination versus monotherapy, especially if you're going to combine it with cytotoxic chemotherapy, which has its own toxicity issues.
Okay. I think with that, we're pretty much running out of time. So I want to thank Dr. Patel for joining us today.
Thank you. So thanks for having me.
And I'm going to turn it back over to Irene.
Thank you very much for joining us. And thank you very much for to our sponsor, Taconic. Taconic Biosciences has been a leading provider of research models for over 60 years. If you have any further questions or comments, please email them to pharma@informa.com. Our next Immuno-Oncology webinar sponsored by Taconic is Friday, December 4th at 4 p.m. GMT time. Thanks for all and have a great rest of your day.